part 4. This will be looking at the remainder of the bacterial structures and functions. Before we move on, I just wanted to come back and hit some highlights of our 3 types of bacterial cell walls, because this will be really important on our lecture exam. And you recall it's important we know what type of cell wall a bacterium has, for it helps us in identification, and very importantly, it's going to influence which antibiotics we choose for our patient. So you'll recall in the gram-positive bacteria cell wall, we have a thick layer of peptidoglycan. This is really hydrophilic, so it offers no protection against water-soluble antibiotics. So the antibiotics are just going to diffuse right through. The other thing we want to remember about gram-positive cell walls is the unique lipoteichoic acid. Right? Okay. And gram-positive bacteria will stain purple in the gram stain. We go down to our gram-negative bacterial cell wall, and you want to remember there's this thin layer of peptidoglycan. And when we gram stain gram-negative bacteria, this thin layer of peptidoglycan can't trap the crystal violet iodine inside, and so the cells decolorize. And then we counter stain using red safranin. So gram-negative bacteria are going to stain red or pink after the gram stain. And very importantly, the gram-negative bacteria have this huge advantage of this lipid bilayer, the outer membrane. Don't confuse this with the cell membrane. The outer membrane is part of the cell wall. And this lipid bilayer creates a hydrophobic barrier against water-soluble antibiotics. For antibiotics to cross the outer membrane, they'd have to fit through these little outer membrane porins, these little protein channels. And some antibiotics can't. For example, penicillin can't pass through the outer membrane porins of most gram-negative bacteria. So generally, we wouldn't use penicillin to treat a gram-negative bacterial infection if we didn't know the identity. What we can do instead, though, if we want to use a beta-lactam, they are our safest antibiotics, is use a semi-synthetic beta-lactam like amoxicillin or ampicillin. And we know that amoxicillin and ampicillin have the right shape and charge and size to pass through those outer membrane porins. So we said ampicillin, amoxicillin are broader spectrum than penicillin. We can use ampicillin and amoxicillin against both gram-positive and gram-negative bacteria. Also remember, in the outer membrane of the gram-negative cell wall, we're going to have these toxic lipopolysaccharide molecules right here, right? And so we have a patient with gram-negative bacteria, say, growing in the blood. When the bacteria start to die, the cells disintegrate, they release this lipopolysaccharide, and that causes one of those cytokine storms we talked about, where your patient can die from endotoxic shock. And then down here, the last one, this is the this waxy hydrophobic cell wall of our acid-fast bacteria. For example, Mycobacterium tuberculosis, Mycobacterium leprae. This is the most resistant, the toughest cell wall, right? So you have to choose your antibiotics really carefully. And we want to, I forgot to mention that one of the frontline anti-TB drugs is a drug called isoniazid. And what isoniazid does is it inhibits the synthesis of this waxy mycolic acid. When you're treating TB, you always use combination therapy, and that means you give more than one drug. So a classic frontline anti-TB drug combo would be isoniazid to knock out mycolic acid synthesis, and then you would add rifampin. And we'll learn later that rifampin knocks out bacterial RNA polymerase. Just our little cartoon of the 3 types of bacterial cell walls. And we had discussed how when gram-negative bacteria are growing in our patients in the inappropriate place, like the blood, for example, or the peritoneal cavity, when they die, they release the lipopolysaccharides. And that causes cells of our immune system to release these huge amount of chemical messengers, the cytokines, and that, like a dominoes falling down, can cause death. All right, and then just one more thing with regard to cell walls and antibiotic therapy, we want to remember that some bacteria lack a cell wall, and therefore they lack peptidoglycan. And the one I wanted you to remember for our lecture exam is Mycoplasma. Mycoplasma is a cell wall-less bacterium. It doesn't have a cell wall, doesn't have peptidoglycan. So consequently, if you're treating a patient with a Mycoplasma infection, such as walking pneumonia, so-called walking pneumonia, you would not choose a beta-lactam antibiotic because the Mycoplasma lack the target. So we'll see later we can choose an antibiotic that maybe targets, for example, the 70S ribosomes, and we'll talk about some other antibiotic targets. Archaea don't make peptidoglycans, so again, if we had an archaeal pathogen, we wouldn't use a beta-lactam antibiotic because the archaea lack the peptidoglycan. So now, folks, we're going to look at some other structures. We'll first take a look at some of the structures found on the outer side of the bacterium, and then we'll go inside. So the first topic we're going to discuss here is the Glycocalyx. This is sometimes the outermost layer of the bacterium. It's on top of the cell wall. Many bacteria have these glycocalyces. Some don't, but many do. It literally means a sugar coat because they're often made out of carbohydrates. Right? There are some exceptions. For example, the Glycocalyx or capsule of Bacillus anthracis is made up of polymers of D-amino acids. There's two basic types of glycocalyces depending on how well organized they are and how tightly adherent they are. There's the capsules and the slime layers. And again, folks, just to get you oriented, you'll see this diagram on our lecture exam. You'll have to identify the parts, the structures and functions. So the Glycocalyx, on your cartoon it's labeled as a capsule. You could put /Glycocalyx, is this outermost layer. And again, you might want to color the cytoplasmic membrane cell wall and capsule different colors so it's easy to distinguish them. So let's first talk about the capsule. Capsules are firmly attached to the outside of the bacterium. They're well organized, and their most important job is to prevent phagocytosis by host leukocytes. For example, PMN, neutrophils, or macrophages. And the reason for this, if we take a look down here at our macrophage, here's one of our macrophages, our big eaters. You can see they make these little cytoplasmic extensions, these pseudopods. And the first step in phagocytosis is they need to attach to the invading bacteria, here represented in green. And to do that, the phagocytes have to have surface receptors to bind to something on the surface of the bacterium. And unfortunately, human phagocytes never evolved receptors to bind to the capsule, or usually polysaccharides of bacteria. And thus, the phagocytes, if they can't attach to the bacteria, they can't ingest them, they can't phagocytize them, they can't kill them. And so again, we would say that the capsule then is anti-phagocytic. And we will see with many bacterial pathogens, if they lack the capsule, the host leukocytes, the phagocytes easily kill them, and basically they won't cause disease. These are 3 encapsulated bacterial pathogens, which again, it's the capsule that lets them avoid our immune system and cause disease. Haemophilus influenza, Streptococcus pneumonia, and Neisseria meningitidis. Haemophilus influenza capsular type B is a common cause of middle ear infections, even meningitis in little babies. Luckily, we have a vaccine against it now, the Hib vaccine. Streptococcus pneumonia, as the name suggests, can cause respiratory tract infections, pneumonia, and this is a big problem in us older folks as we age, right? Our immune system starts to fail us. And again, we're really lucky because we have vaccines to protect us against this bacterial pneumonia. And then Neisseria meningitidis is a cousin of Neisseria gonorrhea, and as the name suggests here, Neisseria meningitidis can cause meningitis, really life-threatening. And this is sometimes the target audience for this bacterium is young people like yourselves. And again, they do have a Neisseria meningitidis vaccine. It's often recommended if young folks are going to be in really crowded, stressful conditions, like maybe first year at college in the dorms or entering the military, you'll get vaccinated against meningitis. The slime layer is a glycocalyx that's more loosely organized and loosely attached to the surface of the bacterium. It can help prevent drying or desiccation, as can a capsule. But probably a more important function of the slime layer is it lets the bacteria stick to surfaces in the environment, forming these microbial communities we call biofilms. And biofilms are really important. It's believed that many of the bacterial infections in humans the invading bacteria will form these biofilms, and these are microbes living in a sticky extracellular matrix, which is basically the slime layer. It lets them attach to surfaces in our bodies. We'll sample tooth biofilm plaque that's attached to our teeth, and we'll gram stain it to look at the microbes. The biofilms can develop on artificial surfaces in our body. So, for example, artificial heart valves, indwelling catheters, artificial joints. Those are great sites for biofilms to develop. Or damaged tissues. So, for example, if a person has damage to the heart valves or maybe has had a heart attack, that puts that patient at higher risk for biofilm formation. And especially you want to be worried if you have a patient, say, with a damaged heart who's going to undergo some dental procedure. The bacteria from the mouth can get introduced to the bloodstream, and then they can attach to that damaged heart tissue and cause a biofilm like an endocarditis or a biofilm on the heart. So often dentists will put patients on antibiotics before they do dental procedures for them. And we're going to see a big problem with biofilms caused by pathogenic bacteria is they're 100 to maybe 1,000 times harder to treat than bacteria that are just free floating, which we call planktonic bacteria. So often, let's say it's maybe a Staphylococcus aureus biofilm formed on an indwelling catheter. You have to remove that catheter, remove the foreign body, treat with antibiotics, and then you have to reinsert the catheter because, again, it's really hard to kill bacteria living in biofilms. And again, folks, this is just a follow-up. This will be our lab exercise. One of the oral means in the mouth bacteria that's great at making the sticky Glycocalyx is Streptococcus mutans, and you can stimulate Glycocalyx production by Streptococcus mutans by eating lots of sugar. So the bacteria then they form this sticky Glycocalyx. They stick to our teeth. Other microbes stick to this sticky material. All the microbes start growing, and then many of the microbes will take more sugar that we eat and ferment it, making acids, and then the acids will decalcify our teeth, increasing the chance for dental caries, cavities. And if this biofilm, this plaque, isn't cleaned off occasionally, it'll cause chronic inflammation to our gums and cause really serious periodontal disease. And indeed, folks that don't have good oral or dental health, it's thought some of the bacteria from the biofilm can get introduced into the bloodstream. If you do brush your teeth or you eat, and then these bacteria can colonize other parts of the body and may in fact increase our risk for cardiovascular disease. So the layers of the biofilm are going to protect the bacteria from antibiotics. There's a diffusion barrier. The layers of the biofilm protect against antibodies, immunoglobulins. The layers of the biofilm prevent phagocytic cells from attaching to the bacteria. And often bacteria in the deeper layers of the biofilm are in a kind of resting stage of growth we call stationary phase of growth. And it's for all those reasons that the bacteria living in the biofilm are so much harder to kill than planktonic bacteria. Other structures outside the cell membrane, plasma membrane, these structures are going to look like little filaments, and they're all made out of proteins. So the flagella, the fimbriae, and the pili, they're all made out of protein. So the fimbria here, right? So this fimbria, in the old days, they would call it an attachment pilus. Oops, let me back up here. And another structure used for the bacteria to attach to surfaces in their environment, there'd be a special protein right at the tip called an adhesion that would bind to a complementary molecule receptor, say on another cell. For example, if this bacterium was living in our bladder, it would use its fimbria with its adhesions to bind to the bladder epithelium, or if it's a bacterium living in our intestine, it'll use its fimbria and adhesions to bind to the intestinal epithelial cells. The reason they want to stick is they don't want to get flushed out. And then, oops, go back here. Here's the flagellum. Flagella is singular. Excuse me, I misspoke. Flagellum is singular. Flagella is the plural. And these are structures used in motility. These flagella are going to rotate like a boat propeller and help the bacteria move through a water-rich environment. You don't have to know the differences in flagella between gram-positive and gram-negative bacteria, but do know that there's three basic parts. to bacterial flagella. There's a basal body, and the basal body anchors the flagellum in the cell membrane and cell wall. There's a hook, and then the long filament made out of protein is called the filament. And the flagellum is just a little bit more complicated in gram-negatives. But again, you don't need to know the difference between gram-positives and gram-negative flagella. There's different arrangements of flagella. I won't ask this on lecture exam 1, but you will be asked this later on lab exam. So a bacterium may lack a flagellum, and that's called atrichous. A bacterium can have a single flagellum, that's monotrichous, or they could have a tuft of flagella at one end, that's called lophotrichous. Amphitrichous is a flagellum at both ends, and peritrichous, my favorite, is when the bacterium is covered with flagella. Okay. And again, the flagella, they rotate like boat propellers, and that's what helps the bacteria move. So in lab, if we look at a wet mount of motile bacteria, we'll see that they move in a series of what are called runs and tumbles. So when the flagella rotate in one direction, it causes the bacterium to move forward in a run. And then the bacterium will reverse the direction of flagellar rotation, and that causes the bacterium to spin in space or do little almost like backflips in space. That's called a tumble. And then the bacteria reverses the direction of rotation of the flagellum, and it sets out on another run, and then it'll do another tumble. So they're kind of fun watching them move through space. So why are they doing these runs and tumbles? Well, they are responding to messengers in their environment. So for example, some bacteria have chemoreceptors for chemicals on their cell membranes, and when those specific chemicals bind, there's a complicated mechanism that then will change the direction of flagellar rotation. So when microbes move along a chemical concentration gradient, we call it chemotaxis, right? And positive chemotaxis is when the bacteria move from an area of low chemical to higher chemical concentration. For example, if the chemical was nutrients, right, the bacteria want to go to the highest concentration of nutrients, so that's positive chemotaxis. But if it was a toxic molecule, the bacteria want to move away from the toxin. They're going to move from an area of high to low chemical concentration, and that's called negative chemotaxis. This is a special type of flagellum found in a group of bacteria called spirochetes. And spirochetes are spiral-shaped bacteria, right? And two spirochetes that are pathogens I'd like you to know for our exam are Borrelia burgdorferi, which causes Lyme disease, the tick-borne disease we have here in California, and then Treponema pallidum. This is the bacterium that causes the sexually transmitted infection, syphilis. Now, the endoflagella are really cool. So there's a bundle of flagella that attach to both ends of the bacterial body. So there'd be one tuft of flagella that attach here and then wrap themselves around like the body of the bacterium, and a second tuft of flagella that would attach at the opposite end, and they wrap themselves around the body of the bacterium. That bundle of flagella, those bundles of endoflagella, form an axial filament. The whole thing is covered by a little thin membrane, and that's why these are called endoflagella. When the flagella rotate, it causes the entire spiral bacterium to turn almost like a corkscrew, like if you're using a corkscrew to open up a corked bottle, and that permits them to drill through thick mucus blankets, maybe drill into tissues. And this might help us understand why both Borrelia burgdorferi and Treponema pallidum, they start at the initial site of infection, and then they can spread throughout the body. They can spread to joints, and sadly, in some infections, they can spread to the central nervous system and cause permanent damage. So we want to treat these guys as soon as we can with antibiotics. So in this colored electron micrograph of a bacterium, these long filaments are flagella, but then notice these shorter little filaments here. They've been colored red. This is artificial. So these little short little hair-like protrusions are the protein fimbriae, the attachment fimbriae. And hard to see here, folks, but we can just barely see in this electron micrograph all these little hair-like fimbriae, And again, they're going to be used in an attachment. So here's a little cartoon. Let's pretend this is the lumen of our intestine, and this is the surface of our intestinal epithelial cells. And all of our cells have different molecules on the surface, right? And here's our bacterium that wants to live in the intestine, and here are the fimbriae. And these little balls at the end, that's our cartoon for the special proteins called adhesions. So for this bacterium to stay in the intestine, it wants its adhesion to bind, kind of like lock and key, to this host cell surface receptor. And it's that attachment that will prevent the bacterium from being washed out of the intestines. And over here, this is a favorite. This is a photomicrograph. This is the lumen of the intestine. These are the intestinal bacteria. These are the little, I think, microvilli here. So help me out, A&P folks. This would be the small intestine. I hope I'm right there. And here we can see an intestinal bacterium. And in lecture, hopefully when we blow this up, you can barely make out the little fimbriae that are attaching to the surface of the intestinal epithelial cells. This is just the coolest slide, I tell you. Okay. One way humans can prevent colonization by pathogenic bacteria is that if we can make antibodies against those protein adhesions, the antibodies bind and then block the ability of the bacterium to bind to the surface of our cells, and then that will prevent disease. And this is just our cartoon for that. If we had antibodies, which you might remember usually Y-shaped molecules, I'm just using little V's here. If our antibodies bind to the adhesions, the bacterium can't bind to our cells, and then the bacterium usually gets washed out. We prevent attachment, you prevent disease, you prevent infection. The third hair-like filament, you could argue, is another attachment structure. This is the sex pilus, also known as the F pilus or conjugation pilus. And we'll see this is used in one example of horizontal gene transfer between bacteria called conjugation. So in conjugation, we have two bacteria involved. So we'll have a donor, and the donor has the genes to make the conjugation pilus. And then we'll have a recipient who lacks the genes. And these two bacteria, the donor and the recipient, are going to exchange chemical messengers, and that lets each of them know that an appropriate, we could say, mate is in the environment. So in response, the donor then is going to start to synthesize this long sex pilus, conjugation pilus, made out of little protein subunits. Gets longer and longer and longer, and then at the end there'll be special proteins that bind to surface receptors on the recipient. Once the donor is in physical contact with the recipient, the donor disassembles the pilus, and that makes it shorter, and that draws the recipient into really close physical contact with the donor. And then it's at that point that the donor is going to transfer a copy of DNA, either plasmid DNA or chromosomal DNA, through the base of the conjugation pilus into the recipient, almost like a little hypodermic syringe and needle. The donor injects the recipient with a copy of DNA. And again, we're really worried because this is a great way for antibiotic resistance genes or toxin genes to be shared. Whoops. Sounds like we had a major disaster. So just a second, I'm going to escape and go see what fell down, and I'll be back to finish up.